sort of from start to finish. So the programming right through to using software to answer biological questions. So there are a number of different tools that you can use to conduct bioinformatics, sort of on the command line at undergraduate level. And again, this is a room that is very familiar with that, so I'll just go through it quickly. You have the sort of secure server approach. You give every student a username, a password, and they have access to tools that you have on the server, and they can do their work. You can also use a virtual machine in a similar fashion. The constraints of those are that students don't really get the full experience of sort of computational biology or computer programming because they don't have any administrative access. In that case, they can't sort of download things, install things. So that is one of the limitations there. You can use a USB approach, so give everyone a USB, contains everything they need to do their bioinformatics work, which is great if it's compatible with whatever hardware that they're trying to use it on, which we know there's a huge range of different laptops, different operating systems, and not all of them are going to be compatible. And then there's a laptop approach, which is very expensive. So that's giving every student a laptop that has everything they need on it to do the bioinformatics work. So one of the solutions to this was the sort of early work of this project, the 4273 Pi project, which created an SD card image for a Raspberry Pi computer. And I don't know, I think, are most people familiar or have used a Raspberry Pi or know what a Raspberry Pi is? Some people say no. So it looks like this. It's just a really small computer. And I managed to, just in evidence of how transportable they are, I brought one all the way from Edinburgh, and it made it here safe and sound. This one does have a case on it because we take it to schools, and pupils aren't sometimes, students, anyone could drop it or whatever, but the computer itself is that green board in there. And this is what Daniel used in an undergraduate module, so a final year undergraduate module. And it was unique in that it allowed students sort of full administrative access. So they could download software, they got a chance to program things, as well as answer biological questions using bioinformatics tools. So just to give you an idea of what that looked like, pretty much you set up one of those small computers to a monitor, a keyboard, and a mouse, and you have a fully functioning computer that has a Linux environment on it, and you can run a number of bioinformatics tools. So if it runs on a computer, chances are you'll get it running well on the Raspberry Pi. So that was the beginning of this work and this project, and that was, as I said, carried out by final year undergraduate students, so sort of more senior students at the University of St Andrews. And just to highlight again why the Raspberry Pi was chosen, it's very low cost compared to buying everyone a laptop or kitting everyone's computer rooms with these fancy new computers that can run Linux. I think you can get it for around sort of all in maybe about £60. Don't quote me on that, but it's less than 100, which is really cheap considering what you can do on this. It's transportable, as I've shown. Students have administrator access, so they can have the sort of full experience of installing softwares and creating their own work environment within bioinformatics. And it avoids hardware incompatibility, so you don't run into those issues that you would do with a USB stick. So again, just to highlight the simplicity of the setup, this was actually a photograph taken at school, and I'm going to talk about our secondary school work in a moment. But you simply, just sort of in the very end of the photograph there on the right-hand side, you see the small Raspberry Pi set up using equipment that a school already has in either their library, their classroom, or an IT suite. So it just takes USB ports, maybe an HDMI cable, and you're all connected and ready to go. So this worked out quite well. And then one of the next missions was to make this available to more junior undergraduate students. So whilst the previous module had a number of weeks of courses running through more sophisticated tools and programming, a new course was designed, a new module for first-year biology students at the University of St Andrews. And this was, it used a gene called the Gulo gene that I don't know if you're familiar with, but I'll talk about it a bit more, as a case study, and still incorporated these Raspberry Pi computers. So at this point, I'm going to move on now to talk about our workshops in secondary schools, because essentially what we do in secondary schools is this workshop. So the workshop that was designed for first-year undergraduate students, we now take to senior secondary school or high school students. And the idea behind that was undergraduates, as is quite a common, I guess complaint's not the right word, but an issue when teaching undergraduates is that they haven't really had any experience of bioinformatics, computational biology, and in fact, any form of computational education before they reach university level. So the thinking was that undergraduates, do they really know much more bioinformatics than senior secondary school pupils? You know, or could you take something that works for first-year undergraduates and make it work for final-year high school students? Because like a language, everyone has to begin at the beginning. So these workshops were trialled in secondary school in Scotland, so in Forfar, not far from where I grew up in Dundee, actually. And it was found that in final year, so sort of penultimate and final year students, so that would be the age range of around 16 to 18 years old, students that have chosen to take higher biology or A-level biology, they actually really enjoyed the activity. According to feedback, they found it quite useful. And teachers were pleased to have researchers come in and, you know, provide these resources. So I'll talk more about what the workshop is, but just to give you an example here, this is a tweet, and we're quite lucky that teachers now are very active on Twitter, so we get a lot of interaction, you know, during workshops or after a workshop when they've, when they've had it, and they're saying what the students have done and what it's helped them achieve. Because obviously from the teacher's perspective, they're very focused on the curriculum. So this particular class was actually studying for their prelim, so their preliminary examinations using this workshop. So secondary schools, we've chosen to focus on Scotland because one, we're a small team, so a manageable goal is always the best way forward with these things. And also we have a way in Scotland because the Scottish curriculum actually includes bioinformatics. It's there, it's one of the key areas, and it has to be covered. However, it is essentially just a couple of sentences in a textbook, and this kind of confusing and, I don't know, just taking up space diagram that I think is getting at bioinformatics being interdisciplinary. Which it is, which is great, but it doesn't really inform students of what bioinformatics is. And I think the sort of SQA, so the Scottish Qualifications Authority, what they want in an exam is a student to say that it is the sort of intersection of molecular biology, statistical analysis, and computer studies. And it does cover all those things, but students just learn that sentence and then they don't really go much further with it, and that's for a number of reasons. So we've had a lot of teacher demand for this when the workshops were initially rolled out. You have a range of teachers, so some teachers, as was mentioned in a previous talk, the other keynote this morning, was that teachers that have their most recent degree within the last 5-10 years, say, they've been trained in bioinformatics. But then you also have teachers that have been teaching for a long time, and bioinformatics wasn't a thing when they were doing their science degree. So you've got a wide range of teachers there, and they have to teach this. It's on the course. So they really are out there looking for resources, looking for ways to teach this effectively. So what we did was there's a mailing list called Synapse, and it connects to all biology teachers in Scotland, or it should. They're usually encouraged to sign up to it. And we offered it as a free, which is important, completely free, curriculum-linked workshop that we will come in provide all the resources for, and fit it within the school's schedule. So we got a lot of responses for that, actually. So we're very fortunate. We've never really had any trouble getting teachers engaged, getting pupils engaged. And I'm going to talk about a bit later, about how we prioritize visits and what we're doing to work on that. But first of all, I'd like to talk you through the workshop. So our first workshop is based, as I said, on that first year undergraduate module. And it's called The Power of Computers in Biology. It's a very all-encompassing title there. But it's a workshop for 16 to 18-year-olds. So these students would be studying higher biology, which is equivalent to A-level. I'm not sure how that translates anywhere else, but sort of towards the end of school, the university qualification, entry qualification level. Higher human biology, and advanced higher, which is just sort of one step above the others. And what we do is we ask that students have prior knowledge of mutations in this workshop. So part of their Unit 1 for this qualification is to cover what DNA is and what mutations are. So they have a good idea of things like substitutions, insertions, deletions. They're even familiar with frame shifts, that sort of thing. So then we come in and we bring along our Raspberry Pi computers and a few volunteers usually, depending on the size of the school. And we give a bit of an intro into bioinformatics, but starting very much on the common ground that we have. So the thing about bioinformatics and the things that I certainly found, coming from a background in undergraduate where I had no training in bioinformatics to do my PhD in bioinformatics, it can be terrifying. It looks very complicated, and sometimes it is complicated. And I think it's okay to acknowledge that things are complicated. Everything doesn't need to be completely easy, and that's okay. But what we want to start with is just reminding them how much they do know. So we give a presentation, we get sort of interaction discussing what mutations are, what they tell us, what mutations they know, how that can lead to things like pseudogenes. They're very familiar with that. And then we move on to what bioinformatics is. And we start by just explaining to them the sort of, not shift, maybe shift is strong, but the move towards a very computational approach in biology that they might not be aware of just through their school syllabus. So we show them the NCBI website is the one that we use, that's the database we use, just to show them the extent of the data, like how much is out there. And then we introduce them to BLAST, which is a tool that I think everyone will be familiar with, that everyone's probably used at some point, and is a great entry point, we think, to bioinformatics. So we provide them with a sequence. We call it sequence R. It's unknown. We don't tell them anything about it. And we tell them, in pairs to go off. And at this point, we get them on regular computers, so they're just using the website at first. And they go and do a BLAST search of this sequence that we've given them and tell us what they found. So what they find is that it is a sequence for this gulo gene, which produces a protein called L-galonolactone oxidase, and it's found in the house mouse. So it's more interesting than it seems. That's a horrible name, and they don't like that name. But it's involved in vitamin C production. So then that takes us into another area of their curriculum that they can explore, which is nutrition and health. Because then the next task we set them is to search for this sequence, not against the whole database, but against the human genome, and tell us if humans have a functional gulo gene or not. And actually they cope with this extremely well. So what they do is they do the BLAST search, they get to the alignment, and they're able to spot, because of their prior knowledge and because of the handouts that we give them explaining a bit about the alignment, they're able to spot things like substitutions, but more importantly, they can spot those insertions, those indels. And they come to the conclusion that humans don't have a functional gulo gene, and hence we can't make our own vitamin C. So then that leads to a lot of discussion about, where do we get our vitamin C? They realize from food, and a lot of them actually bring up scurvy because they study that in history. So it kind of makes a good link there between some other subjects that they are doing, so they can now see why it's so important that you get the 5 or 10 or 15 or whatever it is now, fruit and veg a day. So they do that, and that's the intro to what bioinformatics is. But then we move on to the Raspberry Pi and do essentially the same thing again. So that's search for the sequence against the human genome, but we do it on the command line. So we pretty much put everything we need on the Raspberry Pi because we don't want to mess around with school networks and Wi-Fi. It's very protected, and rightly so, but it's quite tricky. So we have all the BLAST software that they need, all the sequences that they need, everything is stored on the Pi. So what they do is I have a video because the thought of setting up a Raspberry Pi and the laptop and making it work was too stressful. So I just recorded a video of what they would do on the Raspberry Pi. So this is the screen that they get. They open it up and it's just a terminal window, which is something usually completely new to students. You'll come across a couple that have used a Raspberry Pi before, maybe in computing classes, but really not many. So it's quite new, and you have to sort of explain what that is. I just don't know where my mouse is. Okay, that should work. Yeah. So then this is the code that the students will type in. I should say that initially we give them a sort of coding 101 type thing. So they'll make a file, I'm just going to pause that actually. They'll make a file, they'll just nano text file, and then they'll find it, then they'll delete it, just to get them comfortable with using the Raspberry Pi. And we're quite flexible about that. There's nothing they can break. It's not connected to the internet, so they go for it. They make a lot of different files, try out things, and then they move on to the bioinformatics part. So they find their sequence R, they open it up, they see it's there, and then we ask them to open, so we use prior knowledge, we know that this gene is on, or this pseudogene, is on human chromosome 8. So that's the chromosome that we provide. We don't put the whole human genome on the Raspberry Pi, just the chromosome. And we ask them to open that, and they see something that looks like this. So it just scrolls through. And we tell them that if they left that running, it would take about 20 minutes at that speed to get through human chromosome 8. Which I think they're quite surprised about. I mean, it still surprises me. I don't think I've quite get my head around when people are talking millions and billions of bases. So having something like that we think is really quite helpful actually, and they're usually quite surprised just how many bases we're talking about here, but also it gives them an understanding of why you might need computers in biology, because we then ask them if they would prefer to do it sort of on a piece of paper, sequence R, and then a book of chromosome 8 and see how they get on. And then they understand, yeah, okay, this is what bioinformatics helps us to do. So then we give them, we provide them with the BLAST command. We try our hardest to really enforce how each step has an equivalent step on the website that they've just done. So when you have this dash query sequence R, we just say that's the same as copying and pasting sequence R and putting it into the search box in BLAST. Just really try and make it clear, because we don't want it to just be that they're copying it out and have no idea what's going on. We want them to see what each step is doing. And actually, once it's broken down, it's a bit easier to understand. So this runs. It shouldn't take too long. It's part of the reason I have pre-recorded it. And then they open their results file, and we just ask them to take a look at their results, and do they see the same thing using this approach as they see using the website? And they do. They find the same mutations. They find that this also shows a pseudogene because of the frame shift. And then students often ask, but why is this an approach that one would use when the website seems so user-friendly and you've got this one sequence and it takes about, it's quicker on the website actually because they're more comfortable with it. So we tried to highlight the sort of pros and cons of both, because the website is very, very much used, not only in training but in research. It's not a bad thing, it's very useful. but it does have its limitations. So we explain that often scientists, researchers are looking at a lot of these sequences and maybe want to have things run, in an automated fashion and not sit there all day copying and pasting. But also this history command, which is very useful because students, pupils at school, part of their learning experience in science is to understand that you have to keep a record of everything you do. So they'll have a lab book whenever they do an experiment. They sort of write down each step, make sure they don't miss anything. And we show them that bioinformatics is just the same. You have to have a workflow. You have to have repeatability there. And this history command allows you to do that, whereas the website, you can be clicking anything and there's no record of really what you've done. So that is the higher workshop. We also, if there's time, we let them search for this sequence on their own animal. So if they have a particular animal that they're interested in, usually their pet, they'll do dog, which is nice because that has a gene and a pseudogene. So you can talk there about duplication and whether or not they think that this animal could still produce vitamin C if it had a pseudogene and a gene. But we also get a lot of wild ones as well, like a lot of deep sea creatures that are not sequenced. But that's a good opportunity to talk then about e-values and query coverage and looking at, reliable data versus unreliable data. So this is just, I'm going to talk about our more formal evaluation later on, but this is just some of the feedback that we've had. I think what we get is that pupils really like that it's a bit different from the school environment where they have to sit down and usually work alone, which I'm not criticizing. That has its merits as well, sitting down and getting on with something. But this is a bit more, we try and cultivate a research vibe so they can work with a partner, they can talk, they can discuss, share answers. No one's going to get in trouble for that. That's perfectly okay. And I think they like that. And teachers as well have been really receptive to it. They have been instrumental in making sure that this is appropriate for all levels as well. We really value their feedback, and it's something that has really helped us build these workshops. So the next slide, I don't think it's an audience that needs convincing. Some people do, but why is there a benefit to bioinformatics at school? So as I mentioned previously, it is good prep for undergraduate. A lot of students come into their first year of a life sciences degree and it hits them like a ton of bricks because they just haven't seen anything like this before. They're not prepared for it. And in turn, that's quite difficult for lecturers, for demonstrators, tutors, etc., to deal with and to make sure that everyone is on the same page. So there's that, but also we're all aware that there is a gender bias in computational subjects, so that spans right from sort of computational biology through to just more general tech jobs. But at school level, we know in Scotland, and I think this actually expands to the UK, but the facts and figures I have are for Scotland at the moment, that biology classes are predominantly female at all levels. So you're looking at about 65% of a biology class in a school in Scotland is female. And that really gives us a way to address a bias that is usually associated with computational subjects in schools. So if you're bringing this into a computer classroom, you're probably going to have a male bias. In fact, you are going to have a male bias of pupils in that classroom. You bring it into a biology classroom, and you're suddenly having access to more female students and sort of exposing them to computer science that they wouldn't normally get. So that's something that really we're very happy with, and we hope that it's very hard to look at long-term impact of these things. It's something that we're working on, but we hope that that will then address the biases that are seen in undergraduate. And yeah, this is actually quite interesting. So in Scotland, the number of female pupils in computer science has actually dropped over the last few years. So instead of 32%, we're now at about 18% in 2018, which is quite astonishing considering the current job market and just the skill set that a computational biologist would have, or someone with computing skills would have. So yeah, it's quite a disappointing trend, but we hope that by going into biology classes, we can in some way influence the numbers. So these are just some more figures, just highlighting what I've really said, that we have this low female uptake of computational subjects, but we're trying to address this by targeting biology classes, which are predominantly female. And this is not just, I mean, we're doing it in Scotland, but this is a very UK-wide, and I think international feel right now that we need to encourage more females to take up not just computational subjects, but STEM in general. Yep, as I touched on earlier, another one of these benefits is that it's really training the next generation of scientists. A lot of the students that are taking higher biology are doing so because they envision themselves going to university or college to do more science, and usually more life science. So giving them a better understanding of biological research, because a lot of our volunteers are PhD students, researchers, postdocs, PIs. We have a huge range of volunteers that come and do the workshop, and as well as delivering the workshop, they also talk about their job, their research, what they do. So it's a sort of public outreach scope there as well. And yeah, prepare students for undergraduate modules. So again, these are just some interactions that we've had with schools. So in one school, when we go to a school, we want to cover the whole school. We don't just do select classes. We also don't do after school or before school, or because we just want to be as inclusive as possible. And we realize that pupils don't, it's not equal for all pupils, that they would be able to turn up to anything extracurricular. So we make it part of the timetable. And we don't discriminate. We don't want teachers to choose their best behaved class. We want to cover everyone. We want to completely reach an entire, like all the students doing higher, all the students doing nationals. So yeah, in one school there were over 100 pupils over a couple of days doing these workshops. So then again, we thought we would push it a little bit further. So then we've come down from final year undergraduate to first year undergraduate to final years at school. And then there was the idea, well, what about even younger than that? Because the school students at higher level really took this and ran with it. And they asked some incredibly insightful questions about the research we do, about personalized medicine, about gene editing, really intelligent, capable young people. Which really, it led us to think that actually maybe at the younger level, they'll be able to do this just fine. So it was actually at my job interview for this job, and Daniel was sat at the table and we were just chatting, and he said, well, what I really want is a workshop for younger pupils, but I just don't know how it would work. I don't really have any idea of what to do. And it just turned out, and it was a bit strange, but I just happened to have a lot of sausage DNA sequences from a previous job. So it does sound weird, but in a previous internship I'd done with The Naked Scientists, which is a BBC radio show and podcast, and they are clothed but are scientists. So it's really good stuff, and I would recommend listening to them. They cover all aspects of science, and they do it well. And one of the programs that I produced was on DNA, and I just wanted to see if we could just DNA barcode a sausage just to see what was in it. Like, because of the horse meat scandal and stuff, it seemed like an interesting thing to do, and there's still a lot of food fraud and food security issues going on, so yeah, it seemed interesting. And we got a lot of results back, so I'm not going to reveal those right now, but this is what we use in our workshop called Food Detective, which is for 13 to 16-year-olds. And these are pupils that are doing National 4 and National 5. So that's the stage before they go on to do Highers. Some of them might not go on to do Highers. So when you get to this stage, doing a science isn't optional, which has its pros and cons. So you're dealing with a wider range of abilities, maybe interest. There's more diversity, I would say, at the national level and sort of kinds of students that you would have involved compared to the higher level. But we wanted to do that because the pro of that is that you're really getting the opportunity, again with this female bias, so that bias is still there, to influence sounds strong, but to show these students some computational biology, to introduce them to topics that they might not have seen before they make those critical choices that will sort of determine where they go at university level. So at this stage, they still have flexibility about whether they want to progress in science or not. So this could change some minds if it turns out that they really like working on computers and they didn't realize that that was a part of biology. Maybe now they will and continue to go on with that. So I'll just briefly talk you through this workshop. It's called Food Detective. It uses DNA barcodes that we got from a real sausage. So it is authentic sequence that they're dealing with, and we tell them that. And they could actually listen to this podcast, in which the butcher is interviewed. The people that are extracting the DNA are interviewed, so they get the sort of timeline of what's gone on. And this time we don't use the Raspberry Pi because we don't want to overwhelm, but we use the website. So they do a blast search again on NCBI with these barcodes, and we ask them what species they find. So it's 100% pork sausage, so they should just find pig. That's their hypothesis. So yeah, we'll see what they get. Just briefly, we take them through the uses of DNA barcoding. Now, at this stage of the school qualification level, bioinformatics is not part of the curriculum. They don't need to know about it. But this workshop reinforces topics that they do need to know about. So for example, they need to have an understanding of DNA, of the basis of why DNA sequencing or information is useful, so about differences between species and differences between individuals. And we hope that this reinforces that. Also, a bit about ecology. So they're very familiar with doing pitfall traps, collecting insects, and those identification keys that you would use. And they're also very familiar with the cons of those approaches in that they're not always useful. So for example, if you have larvae, you often can't tell just by looking at it what species it is. But that's where, for example, DNA barcoding would come in useful. Also industries, we focus on Scotland, so fisheries, huge there. And a lot of fish fraud, believe it or not, goes on. And DNA barcoding is something that would be used there to stop that problem. So moving on, these are the sequences that we give them. It's all available on the website, so if they fancied doing it, like when they got home to show, I don't know, like their friends, or maybe that's hopeful, or like their mum or dad or whoever, like all of this is completely open. Everything is on the website. That's the same for the higher course, obviously if they don't have a Raspberry Pi, but we provide those to schools as well if they ask. So they can do this themselves. And what they get is a table that looks like this. So they find out that, yeah, they do have a lot of pig in there, which one would expect from a pork sausage, but they also have some other animals that they wouldn't necessarily expect. So we reinforce that these are genuine. And I should say at one point we went to a school and the workshop spanned over lunchtime. So we got to the part where they'd filled in the table, not really explained what the results were yet. They went for lunch and it was sausages. And so we're like, but they seemed fine. They just ate them. It was fine. They weren't too shocked. Maybe they sensed that some contamination was going on, which we explained to them. And they're quite familiar with that. So they get the idea of maybe if you touch a surface, mostly because they've watched things like CSI or whatever, and they see that you just need to touch here and take samples, some DNA's there. So what's probably happened, and what has happened, 99% of these reads map to pig. So everything else is contamination, and it's just come from the butcher, and people maybe extracting the DNA. So we hope that this provides them with an idea of what bioinformatics is, but how it's used, and also reinforces what DNA is, because at that level that's quite important. It seems almost too basic, but really, the basis and how they can tell us about different species. And we also then push it a little bit. So you'll notice that we get them to record the e-value. And we do that because we also provide them with some of the reads that don't map so well. So when they blast them, they get these really high e-value results, and these are just sort of things that would be filtered out in quality control. I mean, we don't go into that with them, but that's essentially what they are. And they get things like tree frog and fish and things that it would be hard to explain as contamination. But we use that it's not essential, so we've got a variety of students. Some, just using the BLAST website, identifying the species is enough. Others are a bit more interested in what these e-values are and looking at reliability of results. So we just get them to compare the sort of higher e-values in that second table to the lower ones in the first table, just to give an understanding of what e-value is and reliability in bioinformatics. But it's not something we really force. I mean, the main part is the species identification. So this is genuinely, I think this maybe is a school way of describing that you've learned something, but this is genuinely what a pupil wrote on a feedback form. And I think it is one of the best. Yeah, it just sums it up very well. That's very nice that they felt that way. I've also had some that are like, no, it could be more fun. So I chose to show this one, but because I like that one. But yeah, it seems to be going well. So one of the things that we are really focused on at the moment is our evaluation. So that was a bit about what the workshops are, and I hope you found it interesting. But one of the things that we really need to do is find out if they are effective and what sort of impact they have. And that is, I'm sure, as all of you are aware, quite difficult, especially when you're spending, so I should say these workshops are a couple of hours each. We go in and it's just 2 hours of the school day. And that's the contact that we have with pupils. So effectively evaluating that is something that we're constantly working on. And yeah, maybe after I present, some people will have ideas on how we can improve that. But the first thing I want to show you is our reach across Scotland. So this is a map of Scotland with Shetland all the way at the top there. So we've reached 152 secondary schools. And that's through workshop visits and CPD events. So we go out and do teacher CPDs that help us to reach a greater number of schools. This is around 32% of schools in Scotland. We really want to push to get that to 50% very soon, ideally around 70%. And we think with this combination of school visits and CPDs for teachers, we should be able to do that. So this equates to just under 3,000 pupils, 200 teachers, and we've reached every local authority in Scotland. But one of the things that I mentioned earlier is that we get a lot of requests from teachers. And as a small team with volunteers, we have to really think about how we prioritise visits. So one of the things I've been looking at is the Scottish Index of Multiple Deprivation. And what that is it's a government figure that takes Scotland is split up into regions. These are not regions that you would normally be familiar with. They're data points assigned by the Scottish government. It's not really too relevant. But what they do is that they look at a number of factors within that region and assign the region an index of multiple deprivation decile. So the more deprived an area is, the most deprived areas will be sort of 1. The least deprived areas, 10. And it takes into account a number of different things. So that could be access to hospitals, transport, crime, does take into account education, a number of different factors. And what we wanted to do was see how we sort of fit in, like what schools we were reaching, because obviously it's easy to reach the schools that have more money for trips or that are, centrally based in a big city so they can come to universities and they get these opportunities all the time. But what we really want to do is reach areas and reach pupils that don't have these opportunities. And this is, yeah, this is what I've tried to present here. So each of these dots is a school that we've reached, and the colour is based on their position on that multiple deprivation index. So the darker the red dot, the more deprived, white, least deprived. So from just looking at that, it's hard to really tell what's going on, but it then allowed us to make these very simple histograms where we compared the schools we've reached in Scotland in each decile to the total number of schools in each decile. And just sort of eyeballing it, not even, I mean, I know we've just discussed the importance of stats, but not even incorporating much statistics, just looking at it, you can see that, yeah, there is work that we need to do in those lower deciles to make sure that we are reaching students that would most benefit, probably, from university collaboration with them. So in the next semester, so coming up in September, we have prioritised our visits to the Lothian region because that is a region that sort of missed out last year because we had prioritised sort of Glasgow, the West, and some of the islands, but also within the ISMB 1 to 3. And we really hope that this will allow us to reach students that don't often get the opportunity to visit universities or have interaction with researchers. And this is just a quote from a science fiction novel that Daniel actually introduced me to. And it really sort of hits home when we talk about public engagement or training and opportunities that are available. And it's sort of, to what extent is the material there for anyone who wants and needs it? And to what extent is it sort of there for those that have the information that it's there, the education to obtain that information, and the power to get that education. And that's something that we keep in mind when we're planning events because, you know, being involved in any sort of outreach, any training, any bioinformatics, especially at the school level, children don't have equal sort of opportunities to do these things. They depend on a parent or a carer to introduce them to these things. If it's going to charge, you know, not everyone has money or transport to get to events. So we always try to make it as easy as possible for anyone to access this. So yeah, I haven't actually read this entire book, but apparently it's one of her best works. But I think it's quite, for the time, it's quite a good quote. So moving on to just sort of in the last few minutes talk about what evaluation we're doing. We, at the moment, hand out feedback forms to students and staff. I'm going to focus on the student feedback, so the pupil feedback at the moment, because it's the one we're working on evaluating. And we give them sort of two statements. They found the workshop useful, they found the workshop enjoyable. And then on a Likert scale, from 1 to 3 for nationals, 1 to 5 for hires. This was because when I thought about it, what's the difference really between strongly agree and agree? I just don't know. I find these things hard to interpret, so we decided to simplify it for the nationals, and they go from disagree to neither, to agree, and then for the higher, they just have that additional strongly attached. But what we find is that for both workshops, so national students, we're at about 152 feedback forms from them, hires around about 1,400. And we find that it is predominantly, it looks quite successful, it's indicative of a successful workshop. However, we're aware that these are very open questions. For example, did they find the workshop enjoyable because it got them out of the usual biology class? Is that why they enjoyed it? Is it useful because it got them out of the usual biology class? I think there's a lot to sort of to dig into with effective evaluation. So if anyone has any tips on that, I would be keen to hear them. But that's the quantitative side. And I think, yeah, it seems to be doing well in both terms of how useful it is and how enjoyable it is from the student point of view. Our qualitative evaluation is based on a 2 stars and a wish approach. I don't know if anyone's heard it, but it's very common in Scottish schools, actually, to evaluate anything. So it's something that pupils are already really familiar with. They use 2 stars and a wish all the time. And it's pretty much 2 things they liked, 1 thing that they would improve. So it's nice and simple. It's free text. They just write in the box. And then we're in the middle right now of doing a thematic content analysis on this. And we just want to identify any themes that come up. So things that they particularly enjoyed, and we can work to making sure that those are present in all workshops, or things that they would like to see improved. So again, I'm not going to dwell on this, but the way thematic content analysis works is that you transcribe all of this free text feedback. That's my job. And develop an initial coding framework. So just sort of select codes. So for example, if someone's like, I liked it because I got to work with my friend, you can categorize that as group work, for example. Then you have a final coding framework where you group all these terms, and both Daniel and I will do this independently because obviously this can be quite subjective, so that's how we're going to verify that. And as I said, I'm kind of in the middle of this right now, but some of the emerging themes that are coming out is that the pupils like working in pairs, which is something we didn't anticipate, actually. We thought that they would do this quite a lot in the classroom, but the more we talk with teachers, they actually don't work in pairs that much. So they quite enjoy being able to work with their friend, and we don't assign them a partner. It's sometimes quite tense when we ask them to find a partner. They have to choose between one of their two friends. But yeah, they get to choose who they want to work with, and they like that. The Nationals workshop, because it doesn't use the Raspberry Pi, students now, as standard, just seem to be, certainly in Scotland, seem to be given a tablet or an iPad or a Chromebook provided to each pupil from the school. So they have their own, they're quite familiar with it, and from our point of view, that works perfectly well. They can do a blast search using their iPads, so that means the workshop can be done anywhere. We don't need a computer room anymore. We don't need any special requirements, just they can do it in the classroom. And they like that. They also like the whole class discussion vibe, so a bit more of a research feel. One thing they continually want to improve is the reliability of the website, but don't we all? Like, that's just the way it goes. Sometimes it doesn't work. Sometimes it's a bit slow when everyone's on it, but we just explained that's the nature of how these things work, and they seem to be okay with that. And also they want more sequences to investigate. So some pupils are just a bit quicker, take to it a bit more quickly, and they just want more work to do. So just to finish off, those are our school workshops. Some new ventures that are coming up for us that we're quite excited about in the next few months, actually, is initial teacher training. So while we do CPD and teacher training, we haven't actually provided any training to student teachers yet. But we are going to be delivering our bioinformatics workshops to trainee teachers at Napier University in Edinburgh and also at the University of Glasgow. So we're quite excited about this because student teachers get the opportunity to do a case study where they can do a lesson with a class and write up a report on it, and we're hoping that we can encourage them to do some bioinformatics with students. So yeah, that's our new venture. We're going to continue with our schools workshops and our CPDs, continue to try and cover as much as we can. We also recently, proudly, became members of Goblet, so we have this resource available now. This is just a screenshot of the power of computers in biology. That's the whole workshop in there, plus some additional info. So if anyone wanted to do it, everything that they need is there, and we're hoping that we can do that also for the Nationals workshop as well, although it might be a bit more straightforward. So I just want to finish by thanking Daniel as well for inviting me onto this project, Tom Marr, who's our collaborator in St Andrews, and everyone else, all the volunteers that put in their time and effort to make this project run, the many schools that we have visited and the teachers that organise our visits, and finally, thank you for listening.
Thank you. I really enjoyed hearing about your work. I go into primary schools quite a lot, so I was really interested to hear about this work. Just wondering about increasing your reach. Have you thought about developing any kind of more online resource on this?
Yeah, so that's the Goblet aspect. So getting that information out there is very useful. We also have all of the resources on our website. So what we say is that we've chosen to cover Scotland just because it's manageable, and we have a staff team that can do that. But schools, if they have access to the website, they've got all the sequences on there, and instructions, teacher lesson plans as well. So it would really just take a teacher or an individual to do that. But we have someone that's recently moved to, I'm going to say the wrong country now, I think it's Sweden, and she is now a biology teacher, and she's taken it over there. So yeah, we're trying, but mostly online, not as an official course, but as materials. Yeah.
And of course, one other point, I really enjoyed hearing the sausage anecdote. But yeah, have you sort of thought about doing more on sort of human clinical sort of genomics? That's what seems to have resonated quite a lot with the primary school kids.
Yeah, no, it would be really interesting. I don't come from that sort of background, so I think if we can find someone that could, I think I'd want to do it justice. So maybe someone that can advise on the best.
Don't want to give myself a job here.
So, you know, if that's...
One of the guys who covered that Terry has also put online is about the study of a clinical study.
Mm-hmm.
Diabetes, one variation, and all what happens when you do the bioinformatics analysis, and that's for kids also. It's something that has been done in Switzerland, and Terry has made a guide for that.
So one of the questions I got asked recently at primary school children was, if I have a disability, how does it affect my DNA? I just thought that was brilliant from just primary age kids.
Yeah,
Thank
they
you.
ask amazing questions.
Yeah, they do.
Actually, yeah.
Amazing talk, thank you.
Thank you.
I have a few questions. First of all, how much of your time have you devoted to doing that?
So me personally?
Yes.
So this is my job. This is my full-time work.
Full-time doing
full-time on this project, yeah.
And the other question was like, how much have you been integrated into the local teaching with the teachers? I mean, visiting so many different schools, isn't it also important to be in personal contact with the teachers locally?
Yeah, so that's a really good point that I completely forgot to mention. So we have established connections with a lot of local schools. We have Forfar Academy that we work with quite a lot as a case study. They helped us to develop both the higher and the nationals workshop. But we also have schools in Edinburgh that are being developed as sort of technology hubs. Kind of, they're being given money for technological equipment. One of them is New Battle, that's also in Edinburgh. So we're aiming to work closely with them in terms of trying to facilitate CPDs and then helping us design workshops that would be useful.
And last question, how difficult was it to align all your activities with their prior knowledge and the overall teaching schedule?
So I think it works quite well because teachers have always been very helpful. So once we worked out what was in Unit 1 of the higher biology or higher human biology, where they know about mutations, they know about DNA, then we can go in. We know that's usually roughly finished around September time. They've covered that. So we just go in at a time when they have that knowledge. Thankfully, schools tend to be quite similar in how they approach things. They all, believe it or not, they can do the units in any order. They usually all choose to do Unit 1 first, which gets them prepped for our workshop, which is good. And Nationals is a bit more open, just because it's not curriculum linked per se.
So we're probably all aware after this morning of the criticisms of sort of parachute workshops and their effectiveness. What plans do you have to provide sort of follow-up so that the staff in schools can either can deliver follow-up and return back to the concepts that you've raised with the students in these workshops?
Yeah. So one of the things that we've done a lot actually is teacher CPD follow-up. That was the best place to start for us, because we could then you give the teacher the CPD event, they come to the CPD event, and then the aim of that is that then they take what they've learned and then provide the workshop to their class year on year. So we have evidence from sort of follow-up teacher interviews that we do, case study links with the schools I've just mentioned that teachers are implementing this and that it's a good teacher training tool. In terms of pupils, there's more complexity there, because trying to keep track of a pupil from school throughout university is actually a huge data protection thing as well. We can't just sort of get emails, do follow-ups a little bit later, or ask for, you know, any data about where students have gone to be released. But again, we're thinking that the sort of best line there to figure out if a workshop is useful is through teachers, and then hopefully figure out a bit more about pupils as we go.
Okay, I wasn't thinking so much of the follow-up of the students learning as such as actually returning to the subjects in terms of reteaching later on. How can you empower the teachers to do that? I think you've covered that with your teacher CPD.
Oh, yeah, okay, yeah.
But I'd also point you, because you're almost local, to the TILE network in Dundee. That's the Teaching Innovation and Learning Enhancement Network, which you may find very useful.
Yeah, thank you very much. I'll check that out.
David, can you start?
Cool. Yeah, so that was a really interesting talk. So just something that came to mind, especially when you pointed out that, so one, you've covered schools all across Scotland, including a lot of the Highlands and Islands, which are sort of very remote, very small numbers of students. But even in sort of central Scotland, a lot of schools are incredibly deprived and don't really have many resources. And something I certainly remember, especially when I was doing hires and especially advanced hires, is a lot of schools simply can't offer a lot of subjects at higher and advanced higher level.
Yep.
Either because they don't have someone who can teach it, they don't have the resources, they don't have enough students. So often if students want to do these subjects, they have to travel a lot to other schools or to colleges, and often aren't able to attend all of the classes because of timetable clashes between the schools. So I was wondering if that was something that had affected your ability to put on these workshops reliably in those sorts of schools, and how you might get around that.
Yeah, so one of the things is actually, thank you. Yeah, so one of the things is we are willing to do visits if there are 2 pupils, 3 pupils. We don't have a minimum number, so if the school can facilitate us, then we will go to that school. Alternatively, if there is another school nearby, we'll organise a collaborative event. So the smaller school will go to the larger school, and we'll just cover more pupils there.
Right, that's really cool.
Yeah.
I wanted to pick up on your suggestions about the way the bash history command is used.
Yes.
You appear to imply that bioinformaticians use the bash history command to keep track of what they've done in a reproducible way, whereas I, as a practicing bioinformatician, use it solely to save keystrokes. And I wondered if there were other situations in which you guys viewed it as necessary and helpful to abridge the truth so that in service of some larger curriculum goal.
Sorry, can you say that last bit again? So
that it was helpful and necessary to abridge the truth in service of some larger curriculum goal.
I don't think so. I mean, history does give you the history of what's been done, and I get it. You as a bioinformatician will go back and use, key up, and they get to the previous command, and that's very useful, and that's something that we tell them. But I think at that level, when you're running a couple of commands like that, and see that you can have your history printed out, I think that is truthful. And is useful for bioinformaticians at that level. But obviously, because we have other researchers coming in and talking about the work they do, I feel like they do get an insight of maybe other things that they can do on command line. Sort of researcher preferences for tools.
One quick last question.
Mine is society-wide, so can we do 2 quick questions? Okay. Is that okay?
Okay, a quick one. So great talk, but so you go to classes where the prevailing students are female, right? And
Usually, I mean, that's not something we choose. That's the
then
nature.
You do some not programming, but you use the terminal and so on, right? Do you have any follow-up? Is then the interest of these students sparked such that they would enroll into computer science courses after your visits?
Yeah, so it's a great question, and it's one we're very interested in. It's just very hard to track, sort of on a per pupil basis, sort of like, were they thinking about doing computer science before we came? Are they thinking about it now? But what we do know is that the links that we've established with some schools, the schools have now got Raspberry Pis. We've either loaned them or the computing department has bought them, and they implement these in other classes that they're doing. So I think if that's the case, then students will be more familiar with Raspberry Pi, more familiar with computing, and then hopefully one would assume that it does encourage at least a certain number of pupils to pursue this later on. But there's no, it's really hard to track on an individual pupil level.
It's me, sorry. Thank you so much. It was a really great talk, and I will follow up with you more afterwards.
Yeah, thank you.
Everyone in the room seemed really, really interested, and I just want to give everyone a quick. ISCB has been piloting a youth bioinformatics symposium in the DC area for the last couple of years. It actually was brought to our attention by a very enthusiastic high school student who actually presented a poster in Dublin when he was a sophomore in high school at our conference. So we started this program 2 years ago, and we've been doing it as a one-day symposium, and we're in the process of trying to put together a toolkit that we can put out via the Education Committee and through Goblet, and kind of show of hands who would be interested in doing a similar type program in their cities. We run it for 7th through 12th graders, where we get outreach from various professors at different universities that kind of bring down a course, and I have all the information, but just kind of a show of hands if anyone would be interested in. Okay, so I'm seeing some hands, which is good.
What we can do is that we have those that are interested can discuss with you right after the end of the session.
Yeah, that would be great. I would appreciate that. Yeah, absolutely. I'll just wait right outside the room if anyone wants to come chat with me about it. Great, thank you.
Lots of hands on.
Thank you very much.